alaikum warahmatullah. So dear students, last time we have discussed about the RNA and its types, and in last lecture we have discussed the messenger RNA. Now today we are going to discuss about the one more topic of the molecular genetics, that is transfer RNA or simply tRNA. Dear students, as you know that the non-genetic RNA is of three types. One is messenger RNA, second is transfer RNA, and third is ribosomal RNA. All the three types of RNA plays a significant role in the process of protein synthesis. And now we are going to discuss about the transfer RNA. देखो बेटा, ट्रांसफर आरएनए क्या होता है? द आरएनए that transfers the amino acid from the amino acid pool to the site of protein synthesis is called as tRNA. Site of protein synthesis is ribosome. That means the RNA that transfers the amino acids from the amino acid pool to the ribosome is known as transfer RNA. As you know that the site of the protein synthesis is the ribosomes of the cell. Got it? Now simply the transfer RNA is also known as supernatant RNA or adapter RNA, and it plays a significant role in the process of the protein synthesis. The transfer RNA is the smallest RNA that comprise approximately 63 to 93 base pairs. It is also called as soluble RNA. नीट में अक्सर ये क्वेश्चन पूछा जाता है कि किस आरएनए को सॉल्युबल आरएनए कहा जाता है. द ट्रांसफर आरएनए इज नोन एज सॉल्युबल आरएनए एंड इट कंप्राइज अप्रोक्सीमेटली 10 to 15% of the total RNA present in the cell. Okay, so dear students, ट्रांसफर आरएनए एक ऐसा आरएनए होता है जो अमाइनो एसिड्स को अमाइनो एसिड पूल से ट्रांसफर करता है टू द साइट ऑफ द प्रोटीन सिंथेसिस, दैट इज द राइबोसोम. सेल्स में ट्रांसफर आरएनए का कंसंट्रेशन 10 to 15% ऑफ द टोटल आरएनए होता है. वन इंपॉर्टेंट पॉइंट अबाउट द ट्रांसफर आरएनए, जो भी आपने आज तक आरएनएस पढ़े तो आपने पढ़ा कि आरएनए में जो न्यूक्लियोटाइड्स होते हैं वो न्यूक्लियोटाइड्स उसमें होते हैं नाइट्रोजनस बेसिस एडेनिन, guanine, cytosine and uracil. But in case of the transfer RNA, there are the modified bases. These modified bases includes ribothymidine, pseudouridine, dihydrouridine and inosine. These modified bases protects the transfer RNA from the exonucleases during the folding. जब हम एनालाइज करते हैं जो फर्स्ट फॉर्म्ड ट्रांसफर आरएनए होता है, इसमें एक लीनियर स्ट्रक्चर होता है 5'3', बट दिस इज नॉट द फंक्शनल स्ट्रक्चर ऑफ द टीआरएनए. उसके बाद इस ट्रांसफर आरएनए में फोल्डिंग्स हो जाती है और ये सेकेंडरी एंड टर्सरी स्ट्रक्चर बनाता है. और जब इसमें फोल्डिंग होती है, इसी फोल्डिंग के दौरान ये मॉडिफाइड बेसिस इस ट्रांसफर आरएनए को प्रोटेक्ट करते हैं फ्रॉम द एक्सोन्यूक्लियस. और फोल्डिंग ऐसे होती है कि जो 5' एंड 3' वाला ग्रुप होता है, एंड्स होते हैं, इस तरीके से फोल्डिंग होती है. 5' एंड 3' एंड जो होते हैं दे लाई क्लोज टू ईच अदर, and the functional structure of the transfer RNA resembles with the Cloverleaf structure. And the Cloverleaf structure of the transfer RNA was proposed by Cloverleaf structure of the tRNA was proposed by Hole. This is also an important point that is repeatedly asked in the competitive examinations. The Cloverleaf structure of the transfer RNA was proposed by Hole. So According to the cloverleaf structure of the tRNA, the tRNA has 5 arms and it has 3 loops. One is the acceptor arm, second one is T-Sci arm, third one is variable arm, fourth one is anticodon arm, and fifth one is DR. First loop is called as T-Sci loop, second loop is called as anticodon loop, and third loop is called as D loop. So that means the functional structure of the transfer RNA is the cloverleaf structure, and this cloverleaf structure was proposed by Holle, and this cloverleaf structure has 5 arms and 3 loops. Okay, देखो बेटा, नाउ फर्स्ट आर्म जो होता है ट्रांसफर आरएनए का, इसको कहा जाता है एक्सेप्टर आर्म. एक्सेप्टर आर्म, दिस इज द स्ट्रक्चर ऑफ द ट्रांसफर आरएनए. दिस वन इज कॉल्ड एज द एक्सेप्टर आर्म. एक्सेप्टर आर्म जो होती है, इट्स कंपोज्ड ऑफ 7 बेस पेयर्स. इसमें 7 बेस पेयर्स होते हैं. 3' एंड पे CCA OH ग्रुप होता है, और यहां पर ही अमाइनो एसिड इसके साथ अटैच हो जाता है. So that means the amino acid is accepted by the acceptor arm. The amino acid is picked by the acceptor arm. That's why this arm is called as the acceptor arm. Okay? देखो बेटा, second is called as T-Sci arm. T-Sci arm जो होता है, इसमें 5 base pairs होते हैं. It's composed of the 5 base pairs. It has a loop, and that's called as T-Sci loop. The T-Sci loop attaches with the ribosome during the process of protein synthesis. That is why it's also called as the ribosomal site. यानी प्रोटीन सिंथेसिस के दौरान यहां से ट्रांसफर आरएनए ने अमाइनो एसिड को एक्सेप्ट किया. प्रोटीन सिंथेसिस के दौरान T-Sci loop of the transfer RNA अटैच हो जाता है राइबोसोम के साथ. So it's called as the ribosomal site. Okay. Now, next, third is known as the variable arm. Third arm of the transfer RNA is called as the variable arm. देखो बेटा, the variable arm is composed of 3 to 5 or 13 to 21 base pairs. ध्यान से सुनिएगा. On the basis of the variable arm, transfer RNA have been classified into 2 types. One is called as class first transfer RNA, and second is called as the class second transfer RNA. क्लास फर्स्ट ट्रांसफर आरएनए उस ट्रांसफर आरएनए को कहा जाता है जिसमें आर्म एब्सेंट होती है और खाली इसमें लूप होता है जिसमें 3 to 5 बेस पेयर्स होते हैं. एंड क्लास फर्स्ट ट्रांसफर आरएनए जो है, इट कॉन्स्टिट्यूट्स 75% ऑफ द टोटल टीआरएनए पॉपुलेशन. एंड second is called as class second transfer RNA. class second transfer RNA उस RNA को कहा जाता है जिसमें वेरिएबल आर्म भी होती है और इसमें लूप भी होता है. इट इज कंपोज्ड ऑफ, ये वेरिएबल आर्म है और इसमें लूप भी है. इट इज कंपोज्ड ऑफ 13 to 21 base pairs and found in 25% of the total tRNA population. Got it? Now, it is said that the function of the variable arm is not yet known. However, the length of the variable arm is essential in the recognition of a specific amino-acyl transfer RNA synthetase enzyme. Okay? Now, next is called as the anticodon arm. Anticodon arm is composed of composed of 5 base pairs. It has a loop called as anticodon loop, and it's composed of 7 base pairs. Out of these 7 base pairs, The three nucleotides acts as an anticodon that recognizes the codons on the messenger RNA so that the proteins are synthesized according to the information of the messenger RNA. Okay. Now, next one is called as the D arm. It's composed of 3 to 4 base pairs. It has a loop that's called as the D loop. The D arm and D loop stabilizes the tertiary structure of the transfer RNA, and in addition to that, it is involved in the charging of the transfer RNA, in the charging of the amino acids. So it is also called as enzyme site. That means simply ट्रांसफर आरएनए का जो फंक्शनल स्ट्रक्चर है, इट हैज अ क्लोवर लीफ स्ट्रक्चर, एक्सेप्टर आर्म, टी साइसी लूप, वेरिएबल आर्म, एंटीकोडॉन लूप, डी आर्म एंड डी लूप. Transfer RNA has 5 arms and 3 loops, and it plays a significant role in the synthesis of the proteins. That's why it's also called as the adapter RNA. देखो बेटा, the synthesis of the tRNA is controlled by the 497 genes, and all these genes are located on the all chromosomes except chromosome number 22 and Y chromosome. So this was about the structure of the transfer RNA. Thank you.